This is an overview of an article entitled Hyperacute Monocyte Gene Response Patterns are Associated with Lower Extremity Vein Bypass Graft Failure, published in the March 2018 issue of Circulation, Genomic and Precision Medicine. Surgical bypass grafting is an important intervention in treating lower extremity arterial occlusive disease, but 30 to 50% of grafts fail to remain patent 5 years after surgery, despite concurrent antiplatelet and anticoagulation therapies. Prior studies have suggested that patency failure in lower extremity bypass grafts can be attributed to chronic inflammation, which then leads to intimal hyperplasia and in-graft atherosclerosis. This inflammation has been shown in animal studies to be driven by monocyte recruitment to the graft sites, and monocyte recruitment is thought to drive the pathogenic hyperplastic response. As such, the authors of the present study hypothesized that early transcriptomic response patterns in circulating monocytes are important determinants of long-term vein graft success or failure. To address this hypothesis, the authors recruited 48 patients undergoing lower extremity bypass graft surgeries and profiled circulating monocytes from blood samples collected on post-operative days 1, 7, and 28. These monocytes underwent transcriptomic profiling by microarray to look for patterns of gene expression changes in the post-operative period that would be associated with graft failure. Patency of the grafts was assessed using standard clinical parameters such as ultrasound findings, ankle brachial indices, and physical examination at months 1, 3, 6, and 12 after surgery. When the microarray data were analyzed, the investigators found that there was not one single gene per se that was associated with outcome-dependent differential expression. So they then analyzed their data by grouping genes into 50 time-dependent clusters that then underwent several stringent filtering steps, which are outlined in this figure. After filtering, only 3 clusters remained, and each cluster remained dynamic over time and met significance thresholds for differential expression between failed and successful grafts. In this figure, the authors are showing gene expression trends over time within each cluster for failed and successful grafts. In each cluster, the failed grafts had higher monocyte cluster gene expression levels immediately after surgery, suggestive of a heightened early transcriptomic response. The genes were enriched for pathways relevant to cytokine signaling and cellular mobility. The heightened early response was also validated in a trauma cohort, with complicated trauma, the red dashed line, and bypass failure, the blue solid line, both demonstrating a steeper slope of decline in gene expression compared to reference. The investigators then wanted to examine which regulators are driving the gene expression changes of the 64 genes identified in the previously mentioned final 3 gene clusters. Using ingenuity pathway analysis, they identified 8 upstream regulators, which include HGF, CSF3, VEGF-A, DICER1, AGT, IL-6, STAT3, and MYD88. In silico silencing of each regulator gene individually within their model yielded a wide range of predicted aggregate effects upon the downstream mediator genes. Here, you can see the aggregate phenotype score depicted by color, with red shades indicative of graft failure, while darker blue shades represent graft success. In general, early regulator gene inhibition mostly tended to favor movement towards a more successful phenotype, while one major exception was that the removal of HGF was associated with worse phenotypes over the postoperative month. In the left-hand panel in this figure, you can see that the HGF row, which depicts effective HGF removal from the author's model, becomes progressively more red as the postoperative time grows. The right-hand panel in this figure depicts predicted effects of removing pairs of regulators to assess possible synergistic effects. But the predicted effects were not amplified beyond simply adding the effects of each individual regulator. In conclusion, the authors in this patient-based discovery study have shown that increased early inflammatory responses in circulating monocytes associate with bypass graft failure, a finding they validated in a trauma cohort. Rather than a single gene driving phenotypic changes, they also found that gene clusters drive the response leading to graft failure. On further analysis, 8 key regulators were identified as potentially influencing gene cluster expression, although how the regulators interact with each other in a network to do so remains to be determined as an area for future investigation.